have a first talk and then tea break and then another talk. And the first talk will be given by Prof. Crawford on the Great Wide Open.
Is this on? Can you guys hear me? All right, well, it's a pleasure to be here. I want to thank the organizers for inviting me to come out and talk to you today. So I'm a biologist, so this is going to be pretty heavily biology focused, but for the statisticians in the audience, hopefully I'm going to make clear what this data is that we're generating and why we think it's exciting, and some of the problems that we're facing and where the limitations are. So this next slide is a very just real basic pie chart of the breakdown of the human genome. We've seen this before. A very small percentage, only about 1 to 2% of the human genome codes for genes, and that leaves 98, 99%, the vast majority of the genome with little or unknown function. So we know that hidden within this 98% are gene regulatory elements that control these genes. And so the goal of my research is to figure out where those gene regulatory elements are, and hopefully that'll help us understand how these genes are regulated, or turned on and turned off. So why is this important? Well, it's going to obviously help us understand some very basic yet fundamentally important questions, such as why different parts of our body look and behave so differently. Why are some genes turned on in certain tissues and turned off in others? What happens in development? How do you get a single fertilized cell grow into a very complex being? This is obviously a carefully orchestrated event of genes being switched on and switched off at different time points. There are also a number of diseases that are probably a result of genes being turned on or turned off when they shouldn't be. And understanding gene regulatory elements will probably help us understand more about how different species have evolved. So now that the genomes have been sequenced from many different species, we're finding that at least the coding regions are quite strikingly similar between different species. So there's an idea that that it could be that it's just the fact that that genes are regulated in different ways in these different species. So hopefully we'll be able to understand, by understanding where the gene regulatory elements are, some of these different things. So there's a number of current genomic approaches different individuals are taking to identify these gene regulatory sequences. Elliot Margulies on day one talked about different comparative genomic strategies, and the idea is that the most highly constrained DNA is most likely functional. There's also a method called chromatin immunoprecipitation, which identifies the precise location of different transcription factors across the genome and different histone modifications that are associated with these gene regulatory elements. And the nice thing about this method is that it takes advantage of some fantastic technologies, such as tiled microarrays, which are basically glass slides with millions of oligos on it. You can hybridize this material, and this method is called chip-chip. Or you can use some of the latest sequencing technologies that we've heard about in previous talks, such as Celexa or 454, to directly sequence this material. There's also a number of different computational approaches that people can take. For example, looking for consensus binding motifs. And Aran this morning talked about using computational approaches to identify nucleosome placements. So the method I'm going to talk about today has to do with mapping DNA hypersensitive sites, and I'll talk a little bit more about what that is and how we're using it. But the nice thing about this method is that it is a chromatin-based approach. But the nice thing is that you don't have to know anything about the transcription factors or histone modifications. All you need to know is a genomic sequence to get the data. And I also want to make the point that none of these methods are 100% perfect. And in order for us to understand what truth is, it's probably going to take a combination of all these different approaches. So this slide just reminds us that the human genome is not static. We've got about 6 feet of DNA crammed into our nucleus, and the way it works is that DNA is not naked in our nuclei, but it's in fact packaged into these molecules called nucleosomes, as well as higher order scaffolding and packaging structures. So DNA binding proteins are believed to displace those nucleosomes. And when that happens, it actually performs a certain function. It turns genes on, or turns genes off, or some other, carries on some other function. So we're interested in figuring out where these regions of the genome are. These are regions where nucleosomes have been displaced. So it turns out that there's this enzyme called DNase that pretty much non-specifically cuts up DNA. But if you take intact nuclei that are wrapped up in chromatin, and you just totally broke that. Let's see. Still working?
Yeah.
Okay. Let's try that again. Put it inside the pocket. Try it this way. All right, still working? Still with me? Okay, great. So if you add just a tiny bit of DNase to nuclei, it turns out that this DNase will preferentially cut at these regions where DNA binding proteins have displaced nucleosomes. And these regions that are cut primarily with DNase are called DNase hypersensitive sites. They're hypersensitive to DNase digestion. Whereas the regions that are packaged up in nucleosomes remain relatively resistant. Now, it will cut in this area a little bit, but it primarily cuts in these hypersensitive site regions. So DNase hypersensitive sites have been around for 30 years, and they've been a very classic and it's been a gold standard method at identifying open regions in chromatin. And there's probably been a couple hundred of these hypersensitive sites that have been described in the literature up until, for the last 30 years. And these regions have been shown to be associated with all different types of regulatory regions, including promoters, enhancers, silencers, insulators, locus control regions, and in yeast even meiotic recombination hotspots. So since these hypersensitive sites are regulating gene expression, we wanted to identify where all of these regions are in the genome. So traditionally, the way to identify these hypersensitive sites is by using a southern blot strategy. And the way this works is you take intact nuclei from cells, digest with increasing amounts of DNase. Then you cut with a restriction enzyme, you run it through a gel, you blot it, and you actually probe for a region of interest where you think there might be a hypersensitive site nearby. And when you don't add any DNase, you only see the appearance of this larger parental band. But as you add increasing amounts of DNase, it might be kind of hard to see here, you start seeing a smaller hypersensitive site band, and that allows you to tell approximately where this region of the genome is and where that functional element is. So these southern blots have been the traditional method, and they've been very good at identifying how a number of genes are regulated. However, if we're going to try to identify all hypersensitive sites throughout the entire genome, obviously southern blots might not be the best way to do it. So today in my talk, I'm going to talk about some new high throughput strategies that we've come up to identify very large numbers of these regions. Then I'm going to discuss some of the things we've learned from analyzing this 1% of the genome that some other speakers have talked about. This is the same 1% that make up the ENCODE regions of the genome. And then I'll talk about how we've been scaling up these methods to look at the entire genome, and then finish up with some of the hurdles and roadblocks that we're facing. So these are the two different strategies we've come up with to identify large numbers of hypersensitive sites. We've developed a tiled array-based method, which we call DNase chip, and basically you can use any tiled array platform. We use arrays from this company called Nimblegen, which is based out of Wisconsin. And the nice thing about these arrays is that you can focus in on a targeted region of the genome. Say if you're interested in how just one gene is regulated, or if you've got a family of genes that you're interested in how they're regulated, you can make custom arrays to look at those specific regions of the genome. Or with enough arrays, you can go across the entire genome, and using the Nimblegen platform, I think it takes about 10 arrays. And the resolution of this method is about 300 base pairs, and that might not seem all that great, but this is based on there's a shearing step in our protocol, and it's based on the shearing size of the products. But this isn't all that bad, because hypersensitive sites aren't a single point in the genome. They're about the size of a nucleosome or two. So we think this resolution is actually not too bad. So we've also developed a sequence-based strategy, which we call DNA-seq, and this takes advantage of some of the newer sequencing platforms, these massively parallel sequencing platforms developed by Illumina and 454. And the advantage is that these methods are inherently genome-wide. We'll get sequences from the entire genome. And another interesting advantage is that this method gives you to the base pair resolution. You can tell the exact base pair where DNA is cut. And we didn't think that was going to be all that helpful, but I'll kind of show you some data later on in the talk that actually shows some surprising signals that are hidden within the data that we're generating. So this is the basic strategy of how we make these DNase libraries. We take intact nuclei, again, digest with just a small amount of DNase to preferentially cut at the hypersensitive site. We then ligate short biotinylated linkers. We then sonicate the DNA to just shear up the DNA in the small fragments. And then we enrich for the DNase ends on a streptavidin column. So streptavidin binds to biotin, and it will pull out anything that has ligated to those biotinylated linkers. And so we're in fact just enriching for regions of the genome that are within or surrounding the hypersensitive site regions. We can then attach a second linker onto the sheared end, and we can either label up this material and hybridize it to tiled microarrays, and you can look for peaks of signals here, such as this, or you can again just sequence using strategies such as Selexa, and you can look particularly for sequences that cluster together, because those sequence clusters are representative of true hypersensitive sites. And every once in a while you'll get signals off by themselves, but that's probably a result of just random shearing or random DNA nicking. So, so this slide just shows how just what some of the data looks like. Again, I'm just going to be describing some data looking at 1% of the genome. And these arrays that we're using, they have 385,000 50-mer oligos that are spaced about every 38 bases. We're really only looking at unique regions of the genome. Unfortunately, we can't look at repetitive regions. And we're looking at these 44 ENCODE regions that Elliot discussed in the first day of the talk. And so this corresponds to 1% of the genome. So this is what a representative example of what the data looks like. We like to see peaks of signals that are often found in multiple DNase concentrations. And this is similar to what people see using the traditional southern blot strategy. You like to see a hypersensitive site band in more than one DNase concentration. We've done a lot of quality assessment to show that the data we're generating is quite good. We've done a lot of quantitative PCR validation with a lot of primer sets, and we showed that our sensitivity and specificity are really quite good. And we've also identified a lot of previously identified hypersensitive sites. This is just one example, probably the most well-characterized set of hypersensitive sites make up the globin locus control region, which is about a group of 5 hypersensitive sites upstream of the beta globin genes. They're about 50 kb upstream of the epsilon gene. And so when we do DNase chip using a K562 cell line, which is a erythroid cell type, we can robustly detect all 5 of these hypersensitive sites that make up the locus control region, as well as this one downstream, this 3' one. And we're also picking up promoter regions for these different globin genes. So this is just another example of how these regulatory regions are not only at the promoters, but some of them are quite large distances away from where the genes are. So we're also seeing a very high cross-validation with DNA sequencing, and I'll show you some data regarding that later on in the talk. But basically, both methods are quite good. So this slide is from a paper from the ENCODE consortium that was recently published, and it basically just makes the point that hypersensitive sites are associated with all different types of active chromatin marks, such as different histone modifications, as well as different transcription factor binding sites. So with a single experiment, we can basically identify all different types of regulatory elements. So this slide just shows our analysis of this 1% of the genome using DNase chip from 6 different human cell types. So we've looked at primary human CD4 T cells, just isolated from blood. We've used a B lymphoblastoid line that's one of the HapMap samples, GMO6990. HeLa S3, which is a cervical carcinoma, IMR90, which is a fetal lung fibroblast cell, K562, which is a erythroid cell type, and we've also generated some data from a human embryonic stem cell line called H9. And we've made all this data, and we're continuing to make all this data publicly available on the Santa Cruz Genome Browser. So one thing that we wanted to see is if we've reached saturation after we've looked at 6 cell types in identifying most hypersensitive sites. So to look at this, we looked at the cumulative number of hypersensitive sites that we've identified after adding in additional cell types into the mix. And what we find is that as you add more and more cell types, it starts to level off a little bit, but we think we're ways away from achieving saturation. We think we'll probably have to go out 20, 30, maybe even 40 cell types before this graph really starts to level off. And another interesting point is that after 6 cell types, this corresponds to 8% of the genome. So it could be that a large fraction of the genome is potentially functional. So this slide just shows the comparisons of the hypersensitive sites between these different 6 cell types. Green represents regions that are hypersensitive. White represents regions that are not hypersensitive. And just by comparing this data, the first thing we see is that about 20% of them are ubiquitous hypersensitive sites. They're found in all 6 cell types. They're probably involved in some kind of housekeeping chromatin structure that are probably controlling housekeeping gene expression. So where are these ubiquitous sites? Well, most of them are found at promoter regions, but there's a significant fraction that don't match to promoter regions. So what are they doing? Well, we found that a large number of them bind to this protein called CTCF, which is a protein involved in enhancer blocking activity. So for those of you who aren't familiar with this protein, the way it works is if you have an enhancer on one side and a gene on the other, if there's CTCF binding in between there, you'll actually get blocking of this enhancer from actually working on this gene. And we think that it's probably critical for all cells to have a certain number of these sites to prevent certain enhancers from working on certain genes. So it kind of segregates the genome into different regions. So what about these other 80%? of the hypersensitive sites? Well, they're either cell type specific, they're found in only one of the 6 cell types, or they're common, they're found in 2, 3, 4, or 5 cell types. So we were interested in more about these cell type specific sites because those are probably regions of the genome that are involved in cell type specific gene expression. So to get at what these cell type specific sites might be doing, we did some motif analysis, and what we found is that there were a number of factors that made biological sense. For example, in CD4 specific hypersensitive sites, we found a very strong motif for TAL1, which is a molecule involved in T-cell acute lymphocytic leukemia. In embryonic stem cells, our top candidate motif was for a protein called OCT1, octamer, which is a factor involved in pluripotency. And in K562 cells, which is a erythroid cell type, the top motif was for this protein called GATA, which is a protein involved in erythroid maturation. So some of these do make sense, but there's a number of these motifs that don't make a lot of biological sense. But there's a lot of things we don't know about transcription factors, but now that we have this list, we can actually go in and test to see if any of these factors are in fact acting as regulators for cell type specific gene expression in these different cell types. We've also shown that these cell type specific hypersensitive sites at promoters are associated with cell type specific gene expression. So these are this is just the different cell types, and we just looked at how many of the cell type specific hypersensitive sites found at promoters correlated with cell type specific gene expression, and you see a general correlation here. Now, this isn't a perfect correlation, but again, this is really still looking at 1% of the genome, and there aren't a lot of cell type specific genes that are found within this 1%. So obviously this is going to have to be redone when we start generating larger data sets from the entire genome. So we have started our scaling up process to look at the entire genome, and we're never, at least with current technologies, we're never going to be able to get to 100%. Probably, since there's some very repetitive regions of the genome, probably going to be able to look at 70 to 80% of the genome. And right now we're using both DNA chip and DNA sequencing strategies. The way this works is we're digesting with DNase, attaching linkers, and we're doing both heavy sequencing as well as whole genome tiled arrays. And the sequencing we're doing, we started off using both Solexa and 454 platforms, and what we found is that we were able to get a lot more sequences, about 15 million off of the Solexa platform, whereas for a lot more money we were only able to get about 3.4 million off the 454 machine. So we think for at least this for this experimental procedure, we think Solexa, the Illumina platform, is probably a little bit better. So this is the cell types we've been working on so far. We've only done DNA-seq and DNA-chip on the CD4 cell type. We've done whole genome DNA-chip on these other cell types as well, and we're waiting for sequences to arrive. So this is just a ROC curve analysis, receiver operator characteristic, which just shows the sensitivity and specificity of these different methods. Again, we've seen some of them in different talks today. Data that would correspond to this diagonal line would represent just random noise, but we find that our DNA chip and DNA sequencing data have a very high area under the curves, and we have also combined the data sets between the two platforms, and we find that it only helps just a little bit. So both platforms by themselves are pretty accurate at identifying true hypersensitive sites. So this is just an example of what this whole genome data looks like. Here's a zoomed out region off of the Santa Cruz genome browser, all these different genes here. And we've got our sequencing data, our DNA chip, and we've got a combined data track. And then here's a region that we've zoomed in. And right here are the individual sequences. And we see what we like to look for, again, are clusters of sequences that cluster in close proximity. So these are 150 sequences that just cluster within a couple hundred bases of each other. Now, we do get some sequences that kind of, that don't cluster, and they look like, just background noise. And we originally just thought that was just random shearing or random DNA snicking. But there is some signal in here that I'll talk about in a couple slides. But the main point here is that this sequencing strategy correlates very well with the chip strategy. There are some regions that we only pick up using tiled arrays, and we're not quite sure why that is, but we're going in and validating some of these regions. So one problem that we've been faced with is how to detect these sequence clusters. So when we first started doing this experiment, we were using an older high throughput sequencing strategy. It was before the most recent Illumina and 454 strategies came out. It was a method called MPSS, or massively parallel signature sequencing, that was developed by Sidney Brenner. And so we worked with this company called Lynx Therapeutics that helped generate 400,000 reads for us, which was a lot a ton of reads at the time. And what we found is when we aligned these sequences of the genome, and we just looked at sequences that fell within, say, 100 bases of each other, what we found was that these sequence clusters were very good at predicting true hypersensitive sites. So this dumb clustering strategy that we're using didn't seem to make sense. First of all, I just want to make the point that after sequencing 400,000 reads, what we found is we were only detecting about 20% of all hypersensitive sites. So we knew we were going to have to sequence way deeper to achieve saturation. And fortunately, some of these new sequencing platforms have come online, and we've been able to do that. So how do we now detect these clusters within the genome? Well, we use this Parzan windowing method, which is basically a kernel density estimation. Now, there's different kernels you can use, but we use a Gaussian kernel. And the way this works is that this is just an example of let's just say, here these blue dots represent individual DNA sequences, and essentially we make a Gaussian distribution is placed around each DNA sequence, and the sum of the areas under each Gaussian curve generates this Parzan density estimation, which is in blue right here, and this is basically done in every single base in the genome, and so closely related sequences are actually weighted more, and you see a higher Parzan score. So we're able to do this with our Celexa sequences, and this is just an example of just one region that just shows how this can actually tell us something interesting about these hypersensitive sites. So here's just a cluster of sequences that don't look all that. There might be some substructure in there. But after we do our Parzan estimation, what we find is that this hypersensitive site actually, there's a region of it that's extremely hypersensitive, and then it kind of tails off on the sides. So it turns out that this actually happens quite a lot. And we were interested in if there was something interesting about these peaks that show the maximum scores within our hypersensitive sites. So we wanted to see if there's anything biologically interesting related to where these peaks of each hypersensitive site was. And so we looked at where they were relative to transcription start sites. So what we did was the maximum DNA hypersensitive site score was set at 0. And the first thing we did was we looked at transcription start sites for genes that are expressed at different levels. And what we found was that the maximal hypersensitive site wasn't at the transcription start site, but it was actually 40 base pairs upstream from the TSS. And if we look at different proteins, such as RNA polymerase, we find the same thing. The maximal hypersensitive site is, again, upstream, about 40 bases. But if we look at other factors, such as CTCF sometimes binds at promoter regions, we find that CTCF actually binds on the opposite side of this maximal region, farther away from the transcription start site. So this shows that these hypersensitive sites are actually directional. And once we get more chip-chip data, such as this, we'll probably be able to predict where these different factors are binding within the hypersensitive site region. There's also been studies that have looked at ChIP-seq for different histone modifications. And one thing people have noticed is that there's a dip in histones around the transcription start site. And what we noticed is that this dip isn't at the transcription start site, but it's actually right at the strongest part of the hypersensitive site. So that really is a really strong indicator that that hypersensitive sites truly are nucleosome depleted regions. So where are these hypersensitive sites relative to genes? Well, we find only about 15% of them are found within 2 kb of the first exon. So there's a lot of stuff, a lot of action that's not happening at the promoter regions. About 17% map to the first intron, a small percent map just downstream of the genes, about 22% map to just different introns within the gene, and about 40% map to intergenic regions between genes. And so a lot of these are probably long-range regulatory regions. So another point I just wanted to make up, or not make up, but tell you about was that I'm really not going to make this one up. This is really true. Is that DNA's hypersensitive sites are not binary. They're not either yes or no. There's actually a degree of hypersensitivity. There's regions that are just wide open. We get screaming signal. And then there's regions that are less and less hypersensitive. And then there's background. And so we were interested in where these most hypersensitive site regions are. And what we found is that about 50% of the screamers, the ones that are just wide open, map to promoter regions. And that kind of makes sense because, you know, these promoters, there's probably a lot of factors that are binding at promoters, and the chromatin structure is just probably a little bit more open for DNase to come in and nick. So one thing that I told you that DNA sequencing gives you to the base pair resolution of where DNA actually cut. So one thing we were wondering is, can we detect any substructures within this DNA sequencing data? For example, if we took the distance between sequences that were, say, going apart from each other, would we detect anything about the size of a nucleosome, say 146 bases or maybe twice that? And so we actually did this experiment, and what we found is that, all right, this slide, it's a little bit small for the people in the back, but this is the bases between sequences, and then we looked at the number of sequence tags, and we were expecting to see maybe a hump around 150 bases, and 150 is right here. But instead, we found an oscillation pattern that was a lot smaller. So we were really interested in where this signal was actually coming from and what it was actually telling us. So the next thing we did was we asked, was this oscillation pattern coming from sequences that actually mapped within the hypersensitive site? So we actually did that, and what we found is that basically it flatlines. There's no oscillation signal from sequences that map within hypersensitive sites. Now remember, not all of these sequences come, so DNase isn't a perfect enzyme. It doesn't cut just at the hypersensitive site. There is some cleavage that happens outside. So if we take away the sequences that map within hypersensitive sites, do we detect more of the signal? And in fact, we see a very striking oscillation pattern, and this frequency is about every 10.4 bases, which was a really interesting number because it's known that DNase, so DNA as it wraps around the nucleosome, the minor groove, is exposed every 10 bases. And DNase is an enzyme that preferentially cuts in the minor groove of DNA as it gets exposed about every 10 bases. And we're able to detect that quite robustly. So this is telling us that nucleosomes are very stably placed within the genome, because if they weren't, if they slid around a little bit, you wouldn't detect this signal. It would look, just more like a flat line. So there's a number of other groups that have used different methods to determine precise nucleosome position and phasing. And so we heard from Aran this morning about some of the work he's been doing on identifying a genomic code for nucleosome positioning. And this was a fascinating paper where, just looking at the genome sequence, they were able to accurately predict about 50% of the nucleosomes, which was amazing. And then there's a number of other wet lab experiments that are just starting to come out. And this is just an example of some of the different papers that have come out using chromatin immunoprecipitation for different histones, and then sequencing that material at very high depth, or tiling this data on arrays. And Aran also talked about some of the methods he's using as well to do the same thing. So the problem is that ChIP and ChIP-seq uses antibodies to these histones. They can reveal the approximate nucleosome positions. However, the accuracy of this might be a little bit fuzzy, since micrococcal nuclease isn't perfect. So the way these strategies work is they cut the nuclei with micrococcal nuclease, which cuts in the linker regions between the nucleosomes. Now, micrococcal doesn't just cut here. If you leave it in a little bit longer, it'll start to chew up the DNA as it starts to wrap around the nucleosome. So in essence, what you might be getting data that looks more like this, where you might not have total confidence of where these different nucleosomes are. So we think if we can include possibly our DNA-seq data, in addition to some of the computational predictions that are coming out, such as the things that Aran is working on, can we accurately determine the precise nucleosome position and phasing? So can we detect with certain probabilities that this nucleosome is found exactly here, and this nucleosome is found right here, and so on? So there's another huge problem that we're faced with is, what about repetitive regions of the genome? So we're largely ignoring this fraction of the genome, about 30%. We've got millions of sequences that map more than one time to the genome, and we're pretty much just ignoring that data right now. We're putting it off in a separate bin, and we're only focusing on the unique regions of the genome. But I think there's probably ways that we can be analyzing this data to help get us a better understanding of what's going on in these repetitive regions, because there is increasing evidence that there are some regulatory regions that are found within repetitive regions of the genome. So maybe if a sequence hits multiple times in the genome, we just weight it a little bit differently, or some method like that. There's another major problem that the chromatin people are faced with, is hooking up DNase hypersensitive sites to target genes. So let's just say we have a hypersensitive site here in the genome. What gene is it actually working on? And that's a major problem. It's not always the case that it's working on the nearest gene. There's been a number of cases where it's been found that, say, a hypersensitive right here works on a gene, 5 genes over, or even on a different chromosome. And so this is a major issue that the biologists are faced with. So there are experimental methods that can help you identify, which regulatory region hooks up to which gene, such as some methods called chromatin conformational capture, or higher throughput versions of that. But there are possibly other ways we could think about getting at this question. For example, Manolis talked about using expression data to possibly look for long-range targets for these different regulatory regions. And I think that he's really hitting on something really important. So let's just say we have got a cell type specific hypersensitive site over here, but we don't see any changes in gene expression in different cell types. But over here, we see a cell type specific change in gene expression that correlates with that cell type specific hypersensitive site. And that could give you some ideas of which genes these regions are actually working on. So we're also interested in understanding how gene regulatory elements have evolved. And so one thing that we've noticed is that, and this is work done in collaboration with Elliot Margulies, is that only approximately 50% of our hypersensitive sites are evolutionarily constrained. So there are other groups doing chip-chip studies that are coming up with similar numbers. So it looks like that there are a lot of regions that aren't very highly constrained. And so what is going on here? Are there regions that are, for example, primate specific? Or is there a lot of positive selection that's going on in the genome? And that's why we're getting these answers. So one thing that we're doing to try to tackle this problem is to do these experiments on from multiple species. So for example, we can take the same exact cell type, CD4 T cells, from human or mouse and figure out where their hypersensitive sites are. So we can do DNA's chip on, say, example, human ENCODE arrays or the entire genome now, and we can design custom arrays using orthologous regions, we can identify the hypersensitive sites, and then we can map the mouse hypersensitive sites to the human coordinates, and we found that about 80% of them are able to map to the human genome. And then we can do direct comparisons between the human and mouse data. So we've generated a preliminary data, and it looks like this data is of high quality from both species, indicating that this method can be used on whatever species you have if you have a sequenced genome. So what are we expecting to find? Well, if we take the mouse sequence and align it to the human, there are some regions that align and others that don't. And we expected to find, some hypersensitive sites that correlate right on top of each other. Some that are maybe found in mouse inserted regions, possibly human inserted regions, or possibly regions that can't be aligned very well. But there's probably also regions such as this that are within alignable sequences, but the hypersensitive sites between the two species aren't right on top of each other. So we wanted to look, we didn't want to waste too much time on these regions right here. So for starters, we've eliminated those from the analysis, but instead looked specifically for regions that directly overlapped or did not overlap between the two species. So this is approximately what we found. This is just a Venn diagram that shows what we found. Only about 1/3 of the hypersensitive sites are shared between both species. About 1/3 were only found in the human genome, and 1/3 only found in the mouse genome, which appeared to be, I was definitely surprised. I thought there would be a lot higher overlap. So we wanted to look at this a little bit more closely, and one thing we did was we looked at the percentage of these different categories that mapped to highly conserved sequences. And we found that these shared hypersensitive sites are a lot more highly conserved, whereas the human-only and mouse-only regulatory regions were still conserved, but a little bit less so. But the striking thing is about all of these regions here in white that aren't very highly conserved. So what are they doing? Are these rapidly evolving regions? Are they regions that are experiencing positive selection? We're really interested in looking at those a little bit more closely. So some of the things we're thinking about doing are do these non-overlapping hypersensitive sites, human-only and mouse-only hypersensitive sites, if they don't share sequence, do they share some other kind of characteristic, such as a motif? Or are these non-overlapping hypersensitive sites, are they closely together? Are they closely found in the genome, but just shifted over a little bit? And more so than what you'd expect by just random chance. And then also, we're also interested in understanding more about what happens in more closely related species. So right now we're looking at human and mouse, but what happens when you look at different primate species such as macaque, chimp, and human? And so we're starting to do these experiments from different lines from these 3 different species. And we're wondering, what does this Venn diagram look like? Is it going to converge more, or is it going to stay the same? These are going to be fascinating experiments to do. We're also, as part of the ENCODE consortium, we're going to be working on identifying hypersensitive sites from additional human cell types. So we're going to be looking at anywhere from 20 to 30 different cell types. We can do this from different cell types isolated from human blood. We can activate them in culture to understand more about the immune response and the regulatory elements that are involved in that process. We can take muscle biopsies and isolate myoblasts. Myoblasts are interesting because if you can actually force them to fuse together into these myotubes, which eventually form myofibers. And there's a lot known about the transcription factors and genes that are expressed during this process, but we'll be able to look at the overall chromatin structure changes that occur as well. We also think that these understanding where these hypersensitive sites are could help us with complex diseases. So there's a lot of diseases where there's been regions that have been shown to be associated with a disease, but oftentimes these regions are quite large. And the thinking is there's probably not going to be necessarily a coding mutation, an obvious coding mutation. So here you're going to have a region of association with a disease, and you're going to have lots of non-coding SNPs that are scattered throughout that region. So what if we look specifically at SNPs that overlap hypersensitive sites? Those will be good candidates to see if they have a functional effect and more related to possibly how these diseases come about. We can look at hepatocytes from liver. We've already generated data from undifferentiated stem cells. We can do the same thing from in differentiated stem cells and look to see how chromatin changes occur during this process. This is going to be a little bit challenging because during the differentiation process, it's never a perfect process. You never get 100% of all the cells, say, forming neuronal precursors, for example. So we might have to do some sorting or think about some creative ways to do that. We're also looking at different cancer cell lines and possibly treating these lines with different therapeutics that are known to work through affecting chromatin structure, such as HDAC inhibitors. So this is just an idea. These are just some of the things that we're going to be working on. And so eventually, we're hoping to generate Venn diagrams such as this. Regions in circles represent all the hypersensitive sites. A lot of them are going to be cell type specific, but a number of them are going to be found in every cell type, and those are these housekeeping chromatin structures. And when we add additional cell types to the mix, such as undifferentiated and myoblasts, we can differentiate the stem cells, and we can look to see at the patterns that shift, and we can activate CD4 T cells, as well as make myoblasts into myotubes. And the ultimate goal is, how do changes in chromatin structure dictate gene expression and cell fate decisions? So this is obviously not going to be answered by just looking at DNA's hypersensitive sites. It's going to take a combination of all different and many different data types, such as expression data, chip data for different transcription factors and histone modifications, different computational methods looking for motifs, sequence conservation, SNPs, copy number variants, looking for 3D structures such as this chromatin conformational capture method, which basically detects large loops of DNA within the nucleus. And so this is going to be a major challenge in the future, and the ENCODE consortium is hopefully going to start this process going, but this is piecing all of these different diverse data sets together is going to be quite a challenge in the future. So just to summarize, these hypersensitive sites that we're identifying have the potential to identify all different types of regulatory elements from any cell type, from any species that has a sequenced genome. We can also start identifying putatively functional SNPs that contribute to changes in gene expression, for example, or even possibly contribute to different diseases. We're starting to understand more about how these gene regulatory elements have evolved between different species, and we're gaining a complete parts list, at least the regions that are critical for transcriptional regulation, which is going to be an important component of systems biology. And there's a number of problems that we're facing. Obviously, this data is not binary. There's regions that are more open than others. And identifying weak signal over background is a major challenge that we're facing right now. Also, hypersensitive sites found within repetitive regions or copy number variants, or cancer cell types that have very abnormal karyotypes. These are going to be a little bit more challenging regions to study. Linking the regulatory element to the target gene is a major challenge. Also, achieving saturation. So now that these new Illumina sequencers are out, how deep do you have to go to achieve saturation? And this is a major issue that a lot of us are facing. And it's not 100% clear to me how we're going to answer this, but there's a lot of ideas that are floating around right now. And then what proteins are binding to these regulatory regions? So what's actually at that region right there, and how is those proteins, how are they working together in conjunction with other regulatory elements to regulate gene expression? We're also interested in using this method for nucleosome placement, and possibly even this might even help us understand about higher order chromatin structure, which we really know very little about. So I just want to thank a number of people that helped with a number of these different experiments. First of all, Elliot Margulies from the NIH helped out with a lot of the sequence conservation work. Paul Meltzer and Sean Davis helped with a lot of the initial the chip work. Ron Mackay from the NIH has helped growing embryonic stem cells. A number of individuals from my group are starting to put these different regulatory elements through different functional tests to figure out what are they actually doing. Are they acting as promoters? Are they acting as enhancers or silencers? And we're going to start characterizing a number of these regions to understand them a little bit more fully. I also really want to thank my computational collaborators, Terry Fury and Ziping Wen from Boston University. We're generating millions of data points, and obviously we couldn't do this work without their help, and that's going to continue to be the case. And I also want to thank individuals from Nibelgen, Illumina, and 454 who, without their technologies, we couldn't be doing this type of work. So with that, I'll end, and I'd be happy to take any questions. So thank you. Paul.
Greg, the real screamer hypersensitive sites, are those more likely to be ubiquitous ones, or did you look at that?
We haven't really looked at that. The screamers are more often found at promoters, and ubiquitous sites, the majority of those are found at promoters, but we haven't actually done that intersection, but we should. Yeah, it might tell us more about what these ubiquitous sites are actually made up of and how they're behaving. Yeah, it's a good idea. Ron.
Regarding the intriguing result of the periodicity between the DNA's hypersensitive sites, do you find any sequence preferences also at these periodic signals?
That's a really good question. We haven't looked at that, and we need to. So there's a lot of papers in the literature that have looked at sequence specificities for DNAs, and it doesn't appear to be a very strong sequence preference. It doesn't like to cut within long stretches of the same nucleotide. It likes transitions better, but we should definitely do that to see if there is.
What I mean is that in those specific sequences, it seems like you're arguing, and that might be true, that you're finding specific settings of the DNA's cut site relative to the nucleosome.
Right.
So the nucleosome at that high resolution, by many different papers that have extracted sequences, is sitting in those specific cases around specific dinucleotides, like AAT.
Right.
Dinucleotides are enriched specifically with periodicities of
Correct. Right.
So I imagine in those cases, the DNase is also cutting specifically
at those regions. Right. We should definitely look at that. Yep, that's a good point.
So for these 10 base pair oscillation patterns, so basically John Weidenblatt's data suggests also for linker DNA also have certain patterns. But basically your data suggests that even after 146 base pair, these oscillations still continue, doesn't show any difference, right?
So DNA doesn't really like to cut in the linker regions. It primarily, I think it's too big of an enzyme to fit in there. So micrococcal fits in there a little bit easier to cut. So this oscillation pattern really only lasts for about 14 times, about 140 base pairs or so, which we think is the amount of DNA that's actually wrapped up with the nucleosome. So we can't really say anything about what's going on in the linker regions. Great.
This idea of looking for correlations in the hypersensitive sites with gene expression. So how many sets do you have now where you actually have gene expression data and hypersensitivity from the same cell?
Right. So
we
we've
really need to do that.
Yeah. So we have done that, and I unfortunately didn't show the slide. So hypersensitive sites are definitely correlated with gene expression. There's very few genes where you see expression, but you don't see a hypersensitive site. I mean, handfuls, okay? And we think that that's probably because of repetitive elements that we can't analyze and so on. But on the flip side, there are a number of genes that have a hypersensitive site, but we can't detect gene expression. And it could be that that the genes are just expressed at a very low level that are below the threshold for these Affymetrix expression arrays, or that these genes are poised. I mean, they're off, but they're just ready to be turned on at a moment's notice due to a certain environmental stimulus.
I mean, there's a few known cases of that. Can you rationalize any of those?
Sure. But we just need to do the experiment to show that it's actually happening. So we are starting to do activation studies to look at this a little bit more specifically. But yeah, it's a really good point. Great. Thank you.
So we have continue.